Welcome to another neurobiology video. Today I want to focus on how and why the action potential actually travels along the nerve, down along the axon of the nerve cell. But just as a quick summary, recap from last time, we learned that the action potential is essentially nothing other than a switch in polarity. So we start with the resting potential, which is a negatively charged state of the cell. Usually it's minus 70 millivolts. Then the cell goes into a positive mode, the action potential itself, and then back to the negative. Now this can be split into several phases. You can see it labeled here. We start with the resting potential. Basically the cell is ready to transmit the signal, and then the initial phase here is the beginning of the action potential, where some sodium channels are triggered, but not all of them. And you can see some sodium will enter the cell until we reach basically the trigger point of minus 50 millivolts, and if that is reached here, these minus 50, then all the sodium channels will open. So the voltage dependent sodium channels will also open, which means that a lot of sodium, basically all the available sodium, enters the cell. So we have a lot of positive ions flowing into the nerve cell at once, and that means that the charge will go into the positive, and then the next step happens up here when we reach the, usually it's plus 30 millivolts, so we've actually gone into the positive, and there is a second trigger point. So if we reach this, then the potassium channels get open, which means now the potassium will actually leave the cell. Now the cell is positive, it no longer wants to be in there, so therefore the charge will go straight back down again, and we actually get to a level that's usually lower than before. So we have this hyperpolarization that is usually caused because the potassium channels will remain open a little bit longer, and in the end we go back to the normal resting potential. So at the last phase here, we actually need to remove the sodium that's entered the cell and put that back out, and we'll have to transfer the potassium that has left the cell, and this is of course the job of the sodium potassium pump that we've already learned about in the first video, which is very active here. Now it's important to keep in mind that this is just a quick status, and you can see down here just how quick that is. We're talking 4 milliseconds until this process has passed. So that means it can potentially happen about 250 times per second. So it's an incredibly fast process, and it doesn't happen to the entire cell. It only happens to a small part of the cell. So we're only talking, might have an action potential right here at the axon hillock, or somewhere up here in a dendrite, or down here close to the synapses. So basically the axon and the action potential will move through the nerve cell. So it'll enter somewhere, coming in with the synapse from a cell. So it enters the dendrite, then enters the soma of the cell, and in fact we might have multiple action potentials entering at the same time, and they will then move towards the axon hillock. And here basically it's almost like a mathematical computation. Depending on how much the charge of the action hillock changes, it'll then pass on the action potential, or if it's not strong enough it won't, but let's say it does, and then that means it'll travel alongside the length of the axon down towards the synapse, where it then connects to either a nerve cell or a muscle. So the action potential is something that happens briefly and then travels through the entire cell. So So let's have a look at the quick animation where we can see this quite well. So we're zooming into the brain here, we see the different neurons, and the color here should indicate the action potential actually racing along the axon here. So these little dots represent the sodium, and you can see how the sodium quickly enters the cell and then leaves it again, and that should symbolize essentially the action potential traveling along the length of our nerves. So the real question is, why does this happen? Why doesn't it just stay where it is? And that's where we have to look at another bit of chemistry. So just as a reminder, the action potential is of course nothing other than sodium ions, so Na plus, that has entered the cell. So the sodium has come in here, which means we have now reached the action potential. In comparison, the yellow areas here, these basically are ahead of the action potential, so this is still in the resting potential, everything that's yellow. And the purple area on the left here, these are the areas that are still refractory, so they cannot yet process a new action potential. So these are refractory areas. So why does it move? The sodium that has entered the cell essentially will just behave like any chemical, and it just diffuses. So basically the sodium starts to spread out inside the cell, and as it does so, because it's positively charged, it'll carry its positive charge to other areas in the cell, which means they will start to drift from the -70 that we have as a standard towards the -50, which is the trigger point where we cause a new action potential. And as soon as this happens in a local area, so if we get to -50 here, then basically the action potential will move along. And then the action potential will happen at the next location, so then it is here. And now we have more sodium that is drifting around, because a lot of sodium now obviously enters the cell from both sides. So we've got the sodium coming in, which means it then starts to drift and move around the cell in all directions and cause a new action potential at the next location. Now you might think, well, why does the sodium only drift in one direction? And of course it doesn't. It actually drifts both forwards and backwards. But as a matter of fact, the action potential will only ever move forward. And that is because right after the action potential is passed, while the cell is still in this hyperpolarization, so it's still in the refractory phase. So if you go back in this area here, just after the action potential has passed, it cannot yet trigger a second action potential because the sodium channels actually are blocked entirely. So that means in that short time frame, a new action potential can't be triggered. So the new ones only happen here in front. So basically what we observe in practice is that the action potential will move into those directions that haven't had it yet. And even though some sodium will drift backwards also, it won't actually cause or have any effect in the areas where it's just passed, because chemically they're not ready yet. In theory, of course, after a while, so basically after another millisecond or so, the action potential has moved on, so it's now in the third location up here, and it'll continue to go forward to cause more action potentials over here next. Now in theory, you think, well, if this diffuses, why doesn't it eventually get back to the new yellow area? So we can see after 2 milliseconds, why doesn't it just cause a an action potential somewhere back here, because as you can see, those yellow areas that we have here, they are now back in the resting potential, and they would in fact be ready to have another action potential. But essentially, because just the laws of diffusion means if one or two little sodium ions actually make it, in practice it's more than one or two ions, but the concentration will be so low that basically it doesn't actually do anything, because the further away you are from the original action potential, the lower the actual concentration. So that means after 2 milliseconds or so, there are not enough ions to actually do anything. So the answer why does the action potential move along the axon is that the sodium will diffuse inside the axon, and it has some kind of domino effect. So basically, as it moves along, it causes a new action potential from one area to the next. So basically, it goes from here to here to here to here to here to here to here, etc. And it has all these chemical changes. So new sodium ions, new sodium channels open, sodium comes in. So it basically has like a domino effect racing down the entire axon, racing down the axon. The speed of this, we can actually measure it or calculate it. That is about 5 millimeters per millisecond. So if you convert this, that equates to about 5,000 millimeters per second. So that is 5 meters per second. And 5 meters per second is about 18 kilometers an hour. So that's quite fast. That's the speed of a person running. But in fact, it's not fast enough for many of our applications. So we have, especially us mammals, have a trick to make this much faster, because what we've described here is what we call just continuous transmission, continuous. But in fact, there is a special case that we can't see in all mammals, but we can see it in us humans, and can't see in all animals, but we can see it in humans and mammals, and that is what we call saltatory nerve transmission. So if you remember correctly, the nerve cell is wrapped in these Schwann cells. So we can see that the axon itself is actually only in the middle here, and it only surfaces in a few locations, and all the rest is wrapped in these sort of packaging cells that usually are covering the entire axon. You can also see it here, these little, in this case the yellow, so-called Schwann cells. So all we can see, or the only areas where the axon is actually exposed, is in between these Schwann cells. So we've got these little nodes here where the axon is exposed in only a few locations. Axon exposed. And this has a big benefit. So what that actually means is as the action potential will not just continuously travel the entire length of the axon, but it'll in fact jump, and that's where the name comes from. We call this the saltatory transmission, and saltatory comes saltatory. In Italian, saltare means to jump, So that's exactly what it does. The signal in fact jumps from one node to the next. So the action potential is here 1 second, and then rather than drifting slowly and causing like action potential here, it jumps to the next location, and then it causes a new action potential. So then of course we have more sodium entering the cell goes positive, and the sodium will again drift. It does the same thing, so the chemistry hasn't exactly changed, but the difference is it now doesn't cause an action potential just here and then another one just here, but it'll drift a little bit longer, but the distance between these nodes is just enough that basically some sodium will get all the way across to the next node, which means then it'll cause a new action potential here. If the distance were any longer, then basically the concentration of sodium wouldn't be high enough and the whole system would break down. And if it were shorter, it would just take longer. So if they're in the perfect distance, what we can see is that the action potential will then just jump from one node to the next, and this will speed up the entire system. So we see this nicely animated here. We've got the continuous transmission on the left, and we've got the saltatory one on the right. And here, this is about 10 times faster. So if we've calculated before that it was roughly 18 kilometers an hour, or 5 millimeters per second, per millisecond, we've now got about 50 millimeters per millisecond, and that equates to about 180 kilometers an hour. So that is the speed of a very fast car. So it's measurable. It's not the speed of light or so, but it is very, very fast, and it is much faster than the sort of standard approach, because it has to be. So not all nerve fibers are in fact saltatory. Even in our body, many of them are not. But it is a speciality that we see mostly in invertebrates and mammals. So we've got mostly essentially in animals with large and complicated nervous systems, because here speed really matters. So most invertebrates just have the simple version, and that is much slower. Now, why do we have a faster version? Because we need it. Now you'd be surprised. For example, the pain nerves that transmit pain do not. They, in fact, they are the common ones here, the continuous ones. But quite commonly, the motor neurons, for example, in our body, so motor neurons almost always have the saltatory transmission, so we have this sort of nice little jumping mechanism, so we have it much faster. Now why pain receptors or the pain nerves, why are they not fast? You think, well, it's important to know that I'm about to sort of melt my hand on a hot plate. Well, yes it is, but that's, you know, it's still fairly fast. You'll find out fairly quickly enough to remove your hand. But actually, for certain processes need more speed. So if you think about it, what us humans do, walking upright actually requires a lot of immediate feedback. If you just shift your body weight a little bit, you would fall over quite quickly, and the body has to be able to react incredibly fast in order to shift where the center of gravity is, which muscles you're actually currently using, just to keep your balance. Now, something else that needs to go incredibly fast is your brain. So inside the brain, the interneurons that basically communicate inside the brain, they are usually also saltatory interneurons. In fact, most of the white color in the white area, not the gray part of the brain, the outside is where we say that's where the thinking happens, but the inside, the white bit, the white color actually comes from the myelin, from those wrapping cells, the Schwann cells around the axon. And it's easy to see why. Our brain is very complex. There's millions and millions of connections, and the faster they can communicate, the quicker we can compute, essentially. So if you want to create a nice three-dimensional image, which is what you're doing all the time with basically the plus and minus information that you get from your eyes don't send the full picture, they just send action potentials in various sequences, and your brain will then have to compute an image from that, just like the graphic card in a computer does. And the more computing power you have, the faster this can calculate, the better the picture. So animals with a large complex brain like ourselves need as much speed as we can possibly get, even if this might cost a bit of extra energy, in order to operate our brains. And that is why the saltatory transmission has evolved in those animals with complex brains, so it enables them to actually use them properly. Right, thank you for your attention. Next we will look at how the nerve cells actually communicate. So the synapses between two nerve cells will be the topic of the next video. But so far I just want to thank you for your attention. Until next time.